To start off with, I will first begin by saying that bacteria can be grouped as gram-positive or negative based on their cell envelope. Gram-positive cells, the cell membrane is surrounded by a thick cell wall, versus gram-negative cells have a thin cell wall surrounded by an outer membrane. So first, I'll start off by going over gram-positive cells. These protrusions that you see inside the cell wall are teichoic acids, and those are chains of glycerylphosphate linked by phosphodiester bonds, and they are bound covalently to the murine cell wall. So murine is what the cell wall is actually made out of. Murine is a type of peptidoglycan, a complex polymer of sugars with side chains of amino acids. So as I mentioned, gram-positive is characterized by a thick cell wall, and that is due to the many layers of murine that it has. And again, murine is a type of peptidoglycan. These longer protrusions that you see all the way down, those would be called lipoteichoic acids. I won't really go into much depth about what that is. Moving on, we have our cell membrane. So in the beginning I did mention that gram-positive cells are surrounded by a thick cell wall, so their cell membrane is surrounded by a thick cell wall. So that's their major characteristic. So this cell membrane itself is made of phospholipids, and the phospholipids are actually in a bilayered form, which means that you have a top and bottom layer. It is characterized by a hydrophilic glycerylphosphate head and a hydrophobic tail. So the tail itself would be fatty acid chains, which are linked to the head that I mentioned earlier. We do have these membrane proteins that are embedded within the cell membrane. Those have their separate functions. The importance is to know that in the cell membrane you have these membrane proteins. For gram-negative cells, we do see a thin cell wall surrounded by an outer membrane, and we do see another membrane here. So that's another way that you can remember these two bacteria is that any membranes are going to be made out of phospholipids. So you'll see that head and those fatty acid chains. So for gram-negative, you would see the two membranes and then the thin cell wall. Versus gram-positive, you would see the thick cell wall and then you see the membrane. It'll become more familiar the more that you start seeing the drawings. So another characteristic that's just for gram-negative bacteria would be the LPS, which is located as the top layer of the outer membrane. That itself is lipopolysaccharide. So in another video, I'll actually go over the whole structure of the lipopolysaccharide, but for now, it's just important to note that that's the outer leaflet of the membrane of only gram-negative. Pretty much it's just the outer membrane, thin cell wall, and then this other inner membrane. And then same as the other gram-positive, you do see these membrane proteins, which all have their functions. However, with gram-negative, you see porins, which you didn't see in gram-positive. You see these porins on the outer membrane, which also function with transport inside cells and facilitating other functions. Something else that I can add is that the cell wall is used for structural support, and it functions to anchor molecules, and that usually helps change the chemistry of the cell to influence transport and other cell functions. Another thing I can actually go over is why gram-positive stains purple and gram-negative stains pink. The reason that gram-positive actually stains purple is because their thick cell wall retains a complex of a purple dye and iodine after an alcohol wash. It would pretty much mean that the gram-positive retain the purple dye because of the thick cell wall, so that murine is able to capture that complex from the dye, so it stains purple under the slide. Gram-negative, on the other hand, doesn't have that thick cell wall that the gram-positive does. So you do see this outer membrane. So even more of a barrier for the dye to actually get into the cell wall, which is how the staining is actually performed. So the gram-negative, on the other hand, do not retain the complex. They become translucent and can actually be counterstained with a red dye. And that's actually the reason why they are pink or red under the slide. So that would be the recap for gram-negative and gram-positive bacteria. So if you have any comments or questions, please just comment below. Thank you.